So I just want to start the meeting by acknowledging the Wurundjeri people of the Kulin Nation, who are the traditional custodian of the land where the University of Melbourne sits, and pay my respect to the elders past, present, and emerging. So it's a pleasure for me to welcome you today. First, I'll introduce myself. I'm Fred Holland. I'm the Deputy Head of Department in the Department of Clinical Pathology, and I am in charge of all the teaching and graduate research training activities for the department. So today is the first time we have to perform such an event online. So please bear with us. Hopefully you'll get the information and tips that you need. You'll have a view of what the research projects that are offered in the department are as well. And obviously feel free to ask questions at the end. So if I now move on to the slide, I guess the first slide that I wanted to, show is just a housekeeping slide. Again, reminding you that there is a chat function, obviously, as you would know in Zoom. Please use the chat function to ask questions if you have questions arising during the presentation. I will try and minimize responses during the presentations. We'll keep these for the end, but I will go through them at the time of question time. Please make sure, and you have already, thank you, that you're putting your audio on mute throughout the talk to minimize the interference. And please make sure that you note a link which will come again at the end of the presentations as well. If you are interested in any of our projects, you will find all the details on the website. I assume if you're here today that you know what honours is, so I'm not going to spend too much time on this particular slide. I guess the point that I just wanted to make, and it's the same for the masters, is really that it provides you with a fantastic opportunity to get acquainted with research by working closely with the senior researcher in their group, learning techniques, but it's really an insight about what your life could be like as a researcher if you end up following the graduate research pathways. It is also a great preparation for other type of graduate level courses, including some students actually go to medicine or such courses afterwards. I think it has a lot of advantages, and I really engage you to embrace that honours year or those 2 years of masters. More specifically, obviously, the department is one of 19 disciplines in the faculty that are offering an honours program, and we are also hosting master's students. So I'll describe both courses briefly so that you're clear on what to expect depending on which one of the two programs you're following. In the context of honours, you'll be expected to complete 100 points throughout the 1 year, and this will be through first a common subject to the most students in the medical, in the MDHS faculty, which is called the Intro to Biomedical Research. There are equivalent subjects that are accepted as well, but this is probably the most commonly taken by students. Biomed 4001, which is worth 12.5 points. You'll have to undertake an advanced discipline coursework subject, and I'll talk about ours in a few minutes. And the first semester is also when you start your research project part 1, which is worth 24.5 points for this first semester. The second semester is really dedicated to your research activities. This is really common to most honours courses. And this is part 2 of the research project, which is worth 50 points. In the context of the Masters of Biomedical Science, for which we're also taking projects, it's a 2-year degree. And so therefore you have to accumulate 200 credit points over those 2 years, which consist of 50 credit points worth of discipline subjects, 25 credit points of professional skills subjects, and 125 points worth of research with an oral presentation and a thesis to submit at the end of the 2 years. Again, you'll have to undertake the same introductory subject that honours students have to undertake, which is the Introduction to Biomedical Research, generally taken at the beginning of year 1. And then there is just an example here of a list of professional skill subjects that you can choose from. So it's very much an a la carte system where you'll be allowed and able to choose from a great variety of subjects. Effectively, if you are actually coming up with another idea of a subject yourself, you can put it past the master's admin team, and they will tell you whether that subject could also be accredited for the master's. This is just an example for you in terms of how to organize that master's. Again, I said it's an a la carte type system, so you can play a little bit with the different subjects and when you are effectively doing them. So I've taken two examples. This is an example where there's a kind of balanced distribution between your coursework and your research. You can see here that first semester, at the very start, you'll take the biomed 4.1 subject, but you can have coursework dispatched again at 12.5 in semester 1, a couple of coursework, also potentially your 1 here in semester 2, and your research then makes up for the rest of the 50 points that you'll be doing every semester. So you can see here a balanced distribution between the two. You can see here another way you can organize your masters, which is to actually pile up quite a bit of the coursework early on, particularly during year 1, minimize the research points, and then have a very heavy going research second year, basically. And this will suit some of you, for example, who have ethics proposals to put up during the first semester, whereby you could use those 12.5 points to start writing up your literature review and preparing ethics. So really, it's something to be discussed with your supervisors. I think something that's not on the slides, which is an interesting, I think, point for you all to think about when you decide whether to go for honors or masters, is if you are really envisaging seriously to take up graduate research in the form of a PhD afterwards, and particularly looking to potentially have a PhD scholarship. As you would probably know, excuse me, the PhD scholarships scores are designed basically on your 2 previous years at the university, or actually for external students as well. What this means is that if you are doing an honors degree, what will count towards your PhD scholarship score is the honors year plus your 3rd year of undergraduate of your bachelor's. So if you have had extremely good marks in the 3rd year of your bachelor's degree, I think that's a very good reason to go towards honors in year 4. However, if your year 3 marks haven't been maybe as high as you would have hoped, I'd really sort of encourage you to consider potentially the master's, because effectively the master's being run over 2 years. It's the 2 years of the master's degree only that will count towards your scholarship score. And you could then, hopefully, if you are really thriving through the research masters, you will be able to have a much higher score than you might have had otherwise if you'd kept your 3rd year mark. So food for thoughts in terms of your strategy as well, depending on what you want to do. In terms of the prospects and the future career, a honors or master's degree in the Department of Clinical Pathology really leads to a very broad distribution and array of careers. You can see here a list of those. Important ones are obviously working potentially, besides the research pathways, working in diagnostic laboratories, the medical scientific communication fields, public health policy, forensic pathology as well as the medical legal industry too, which are linked. Obviously, research pathways and other graduate courses such as medicine or allied health courses are really common sort of outcomes for our students too. So you're really well prepared, and we're trying to prepare you really well with learning outcomes that will serve you beyond the simple fact of doing research in clinical pathology. The requirements, again, you will know this, and I won't spend too much time. We're looking for usually an overall weighted average mark or WAM of at least 65. These are equivalents for your University of Melbourne internal applicants or external applicants. This is very similar for the masters as well. It is a very peculiar year, and so there's been discussions about the WAMs on this year. So I think that the best will be for you, if you are around that mark, to contact me as a coordinator to discuss whether, on the one hand, the disciplines and the subjects you have taken in terms of your background in the bachelor's degree, as well as your WAM level, are sufficient for you to be admitted. We'll probably negotiate a little bit more that score in regards to the current issues with the WAM at the University of Melbourne through the COVID-19 pandemics. Now, if we focus on the Department of Clinical Pathology itself for a minute, for those of you who don't know the department, I think it's it's a great department. I'm obviously quite biased, but we are really teaching, first of all, into the flagship course, the Doctor of Medicine, and we are also training students in honors, masters, and PhDs. We are also teaching in the Masters of Cancer Science, which is an online master's degree. And in terms of the research activities, we'll research in the different labs. You'll have a bit of a snapshot coming from the different presentations that we'll briefly show you in a few minutes. Our research focuses really on detecting molecular pathways that are affected in tumors and how we can find and sort of harness, I should say, those weaknesses for a better prognostic of patients and better treatment options for patients with cancer. There are specialist laboratories in our department in cancer cell biology, in DNA analysis of all kinds, in genomics, as well as in bioinformatics and organoid generation and testing. So again, you'll see the list of projects that we'll show you in a few minutes, as well as a few videos that summarize some of those projects. Yep. The department is located within the VITROPOSI building. So you have a picture of the VITROPOSI building at the bottom here. So it's the same building where the other organizations such as the PITAMAC are located. And this is a really fantastic location to work because we can collaborate with other institutes such as the PITAMAC that I just mentioned, as well as our clinician colleagues. And so we are really having a research program that's integrated into the continuum between very basic research to extremely translational research for the benefit of cancer patients. The department is in fact part of all the activities of cancer research at the University of Melbourne are regrouped under a center called the University of Melbourne Centre for Cancer Research, and the Department of Pathology, because its research is really focused on cancer only, is really an integral part of this center. And we are co-located, like I just mentioned, with a number of other partners in the building. Facilities, we have great facilities in the department, including histology FACS, a clinical genomics platform, doing reporting for patients, but also doing research grade genomics. We have single cell sequencing facilities with the 10x genomics system. We have an organoid lab. We have several groups, in fact, working on tumor organoids. We have a biobanking lab, as well as a very large bioinformatics core, which helps the different projects. The department also offers a number of scholarships. This is throughout the continuum from honors to graduate pathways. I'll just cite a few. The J.E. Taylor Honors Award, which is for the best honored students undertaking honors in our department, which is a $2,000 prize, as well as a few PhD scholarships, including a full PhD scholarship called the Margaret and Irene Stewardson Fund Scholarship, which is for up to 3.5 years, as well as some top-up scholarships called either the Beanie or the W.P. Green Scholarship. So you have great opportunities potentially in the department throughout your graduate pathway and your research, beginning of your research career, to be supported internally if you don't get other supports as well. Now I'll move to the honors course more specifically, particularly to sort of explain the structure of that course in more detail, because this is something that will include some coursework, for example, that is really important for and specific for our department. So as I mentioned, the honors course is run throughout the 1 year. In fact, it starts even before the usual start of semester, because the first of the subjects that you will have to undertake is Biomed 4001, which is a faculty-run subject, and this is run as an intensive subject in the 2 last weeks of February. So you'll be starting mid-Feb and have 2 weeks of Biomed 4001. From that point, you'll start the coursework in the department. And so the subject for the coursework in our department is called Path 4002. It's actually a coursework that we've decided to run in collaboration with the other pathology stream, which is run from the Department of Microbiology and Immunology. And the reason for this is because our department's research being really extremely focused on cancer, we wanted to have access to the expertise of other pathologists that work on other areas and other diseases than cancer, to have a more sort of broad and I think more interesting in the end coursework subject. And I think the subject is really great. You'll be assessed in 2 different ways after an initial information session very early in the semester. You'll be assessed in 2 different ways. One is an oral presentation that you will have to give around the first week of April. Please take those dates with a little bit of a grain of salt, because we've been forced really to move around the dates a little bit this year due to the pandemic issues, and so we're not quite sure what the situation will be next year. Hopefully everything will be back to normal, so in which case it will be around the first week of April. And we have a written open book exam called Critical Analysis Assessment, which is generally run in the first week of May. On top of this, you'll have your research project. I'll talk a bit more detail about the coursework in a minute. You'll have your research project, again, spread into 2 semesters, the Path 4 006 subject in first semester 1 and the Path 4 007 subject in semester 2. And for those, you'll have different type of assessments. We'll have a literature review submission, which is a hurdle requirement, so you will not be scored on that. I will be assessing those literature reviews and giving you feedback around the beginning of semester 2. The hurdle is around the 3rd week of May, and you have a introductory oral presentation that is assessed at the end of the 1st week of June generally. In semester 2, you'll have a number of drop-in sessions which I organize, and they've been particularly important this year, organizing them online to touch base with all the students as you go. And this is really an opportunity for you to come and chat with me in case you have any concerns, any questions, and that you prefer to ask them face-to-face with me. You are obviously welcome always to contact me by email or to organize a separate catching up one-to-one discussion if you have any issues that you want to discuss. And usually in a normal year, again, this is really different this year, but I won't go into these details. In a normal year, towards the end of September, we ask you to finish your lab work and to leave generally about 3 to 3.5 weeks for thesis writing. You'll have obviously written most of the introduction for your thesis in the form of a lit review earlier in the semester, earlier in the year, I should say, but you will still have quite a bit of writing to do. So thesis submission is usually around the 3rd week of October for us. And thesis defense seminars are given around the 1st week of November. I should say that in our department, the policy is that you will be scored by 3 examiners for every assessment in the research projects, because we think that it gives a fairer representation of what your performance was. A quick words, this is a very busy slide and I don't need you to remember everything, but really to inform you more as to how we run the coursework. So the coursework, once again, for 0002 is run in collaboration with the Department of Microbiology and Immunology, and particularly with their coordinators, Dr. Theo Mantamadiotis. So Theo and I organize everything. So this is an example. It's going to be different next year, but this is an example of what we ran this year and which was successfully run even though we had to shift to online delivery fairly early on. Importantly, you'll have a series of tutorials, 5 or 6, depending on the years, which will really be focused on developing your skills in critical analysis of research literature. And this is really one of the key learning outcomes for us. So we spend quite a bit of time on this, and you'll be sitting down in groups, generally spread out in groups of 4 or 5 to discuss different parts of research papers, obviously under guidance from our coordinators or other tutors. And so effectively you'll be learning what to look for in research literature, what makes a good manuscript and a poor manuscript. And effectively this is one of the criteria that you will be assessed upon towards the end of semester, as you can see here at the bottom, through an exam. Here was on 5th of May originally this year, which is an oral presentation where you'll be asked to effectively present part of the paper and criticize what you think, or give you a critical analysis of what you think about that particular part of the paper. And a written exam led around mid-May, where you'll also have a number of questions surrounding research literature, where you will be asked to discuss and criticize what's going on in that particular paper. So the tutorials are particularly important and compulsory. There's also a degree of supporting material that we provide in the form of different lectures that will introduce you to a number of the key techniques and skills in clinical pathology and pathology research, which are important for you to then be able to assess that literature properly. So although you are not necessarily assessed directly on the content of the lectures, they are essential for you to actually have enough food for thought in your critical analysis of the different papers that we then expose you to during the tutorials. We also try to organize a number of workshops or little panel discussions about a number of aspects that we think are particularly important in today's clinical pathology research. One of them is systems biology, which has one lecture dedicated to it and a small workshop of computational biology approaches in biomedical research, but also introduction to what is consumer engagement in research, which is becoming more and more a crucial part of what we do. And by consumer engagement, we mean interaction with real patients and discussion with cancer survivors as to why their view of research and medical practice is really important for us to take into consideration. So this is a very dynamic and exciting part of the coursework. So this is to give you really an overall idea of what we try to do. And the coursework, I think, is extremely well received and quite useful, because it comes as a great backup effectively for your research activity and your research projects by giving you additional skills that are useful for those. So now I'll move on to the research projects that are offered in the department for honors and masters. Effectively, you can see here a list of 7. We're actually missing a few that were given to us late, but that you'll be hopefully able to find on the website later on. But so you will have now a series of videos that are going to be played to you by Zoe Stevenson, who is our communications officer, and who has prepared those great videos to support the different projects and give you a flavor of the research that's being done. If any of those projects are of interest to you, you'll find the contact details for the relevant supervisors on the website, and then you'll be able to contact individually those supervisors for further discussions. So I'll get back to you after Zoe has shown those videos.
Ever wondered whether you can catch cancer? Can cancer be contagious? Even though you can't really catch cancer from someone who already has it, there are a lot of cancers that are caused by microorganisms, especially viruses. Perhaps the best example of that is cancer of the cervix. This cancer can essentially be considered a sexually transmitted disease. In fact, the majority of cervical cancers are related to a virus called human papillomavirus, or HPV. My name is Kais Kassem. I'm an associate professor of clinical pathology at the University of Melbourne, and I also work clinically as a surgical pathologist, the type of doctors who diagnose diseases based on the changes at the cell and tissue level. Here I am proposing a research project titled, "Is This Cell Unwell?" evaluating the features of HPV-infected squamous cells of the cervix. Some types of HPV virus can infect the squamous cells of the cervix and cause pre-malignant condition called dysplasia. This is not cancer yet, but it's a step towards cancer. If the viral infection and the dysplasia persist, this will eventually lead to cervical cancer. Now, when a squamous epithelial cells of the cervix is infected with HPV, the cell demonstrates certain characteristic features collectively called koilocytosis. As a pathologist, I observe these changes under the microscope and report their presence or absence in every cervical biopsy. This is very important in establishing whether the HPV infection of the cells has occurred and determining the grade of the abnormality. The presence of HPV infection means this patient may require close follow-up, additional tissue biopsies, or further intervention. Although the microscopic features of HPV infection are well described in literature, there is a number of mimics of koilocytosis. In other words, cells that look like koilocytes but aren't true koilocytes. Currently, there are no definite characteristic criteria to distinguish true koilocytes from their mimics. This study aims at exploring the potential of developing such criteria based on analysis of retrospectively collected data, including molecular HPV studies, but also microscopic analysis of cells and tissue biopsy samples. The topic of cervical precancerous and cancerous conditions is very exciting and super important. The project would suit someone with strong interest in cancer diagnosis and pathology, someone who is perhaps a bit like me, fascinated by the way the cells and tissue look under the microscope. Students with good understanding of basic statistics and keen on being involved in a project that will make a real impact are strongly encouraged to apply.
My name is Lachlan Coyne. I lead the Cancer Bioinformatics Research Group. And this year I'm offering an honours project which is aiming to characterise the repeat variation in cancer genomes. Now, repeats have traditionally been a very difficult part of the human genome to analyse, and there's still quite a lot of the human genome which we know very little about because kind of standard sequencing techniques are too short, and so they can't sequence the very, very long repeats. However, there's recently been development of new technologies, in particular nanopore sequencing technologies, which enable us to generate extremely long reads which can sequence across almost all the repetitive regions in the human genome. And recently we've used this technology to get some sequence data, long-read sequence data from both the primary and the metastatic breast cancer tumour. And we've also been working on algorithms which can look at this data and identify repeat regions which have been amplified relative to what's seen in the reference genome, and also characterise, parts of the human genome which have not been sequenced before. And so this project aims to use and extend those techniques in order to see what differences there are between both the primary tumour and the metastatic tumour in terms of repeat expansions, and also to see variation relative to the normal germline genome. It's a really exciting opportunity to work with cutting edge sequencing data and also address an important medical problem in development of tumours. Thank you.
Hi, I'm Richard Tottill. I'm a group leader at the University of Melbourne Centre for Cancer Research. My group is the Rare Disease Oncogenomics Laboratory. We focus on translational research of rare and less common cancers. One specific area of interest in my lab relates to a disease called cancer of unknown primary, or CUP for short. CUP is a diagnostic conundrum defined as a metastatic cancer where the primary site remains unknown despite extensive clinical investigation. The optimal choice of cancer treatment today is still defined by the cancer cell of origin or tissue of origin. Therefore, CUP tumours represent a significant clinical challenge. In my research, I have been using genomics to assist the diagnosis of CUP patients. This has included the development of gene expression profiling as a diagnostic tool. These gene expression tests are based on the idea that metastatic cancers retain a gene expression profile that is similar to the cell of origin of that cancer. Building a reference of gene expression of known cancer types, we can train a classifier that then can predict the tissue of origin of an unknown tumour sample. These tests work with an accuracy of about 85%, but there's some particular scenarios where the classifier has difficulty. One group of cancers where this is the case includes the pancreatobiliary tumours. These cancers include cholangiocarcinomas that originate from the bile ducts, as well as pancreatic adenocarcinomas. Some cholangiocarcinomas and pancreatic adenocarcinomas have a very similar gene expression profile, meaning that the tissue of origin classification can be more challenging. Our goal is therefore to develop a deeper understanding of the molecular profile of these different pancreatobiliary tumours with a view to developing a diagnostic method. An honours project in my lab will therefore have the following aims. In aim 1, the student will collate published gene expression datasets of pancreatobiliary tumours and other cancers. In aim 2, the student will identify gene sets can distinguish between these cancer types and build a machine learning tool that can be used as a classifier. In aim 3, identifying single genes that can potentially be translated to a histopathology lab as a single gene test. In this project, I envisage that the honours student will receive expert bioinformatics training, experience in cutting-edge molecular methods, as well as substantially increase their knowledge in cancer biology and pathology and genomics. Thank you.
If you want to be involved in an honours project that makes a difference for cancer patients, we hope that you'll be keen to join us for this project that aims to identify biomarkers predicting pancreatic cancer response to targeted drug therapy. You might wonder why studying pancreatic cancer is really an important topic. Well, in fact, there has been little progress in therapies for this cancer over the last 20 to 30 years, meaning that very few treatments are available for patients. The 5-year survival rate for this cancer is around 10% or even less than 10%. And with the increasing incidence rate, it means that this cancer is predicted to be the second leading cause of cancer death worldwide in about 10 to 15 years. In recent years, molecular characterizations have allowed the identification of a large number of genetic alterations that play an important part in the development of this disease. And this has raised great hope in the fact that targeting some of those genetic alterations might actually provide very viable therapeutic options for patients. Yet, one of the things that we're critically lacking at the moment is biomarkers that would be able to tell us and to predict which of the patients would respond or not respond to these targeted therapies. In this honors project, we propose to identify biomarkers to predict the response to a few such targeted drugs. To do so, on the one hand, we would use tumor organoids generated from pancreatic cancer patients to identify a suite of promising targeted drug candidates. On the other hand, we will implement a machine learning approach using a genomic, transcriptomic, and drug response data that we have obtained from these organoids. The aim is to develop clinically meaningful markers that will allow clinicians to match individual patients with drugs that are the most likely to be effective in treating their tumors. This project provides an exciting opportunity for students willing to combine a small amount of laboratory work, in this case, targeted drug assays on tumor organoids, with computational biology and machine learning implementation. The project is co-supervised by myself and my colleague, Professor Lachlan Coyne. We expect that the student would have some degree of previous familiarity with one or more programming languages and a keen ability to learn more computational techniques. So whether you already have experience in laboratory techniques and you want to develop more skills in computational biology, or whether you have done already bioinformatics courses and wish to develop your expertise in that field, and particularly in applied machine learning, why don't you contact us to discuss the project?
Hi everyone, my name is Associate Professor Dan Buchanan. I'm head of the colorectal oncogenomics group in the Department of Clinical Pathology at the University of Melbourne. My lab is situated in level 10 of the Victorian Comprehensive Cancer Centre. So I'm really excited to talk to you today about our honours project that we have listed for you guys to apply to. And it is around, can we determine which colorectal cancers are caused by gut bacteria? So my lab mainly focuses on colorectal cancer, and not the treatment side of things, but more so how to prevent it. And so we're really interested to know, by measuring the bacterial content within tumours, to determine which bacteria are likely to be those that are causing colorectal cancer development. So this is a really exciting project, and a way with which we can take some further steps towards preventing colorectal cancer development by determining the base causes of it. And in this case, this project is looking specifically at genotoxic types of bacteria and their presence in colorectal cancer, and the types of DNA damage that they cause, which is like a fingerprint for tumour development in those particular cancers. So we're really excited about this project. I'm happy to answer any questions you have about around the project. We've got a great team in my lab, a very multidisciplinary and highly skilled group that are really great to work with. And so we're looking for an enthusiastic and keen student to come and take on this project and drive this interesting work on bacterial causes of colorectal cancer and how we can prevent it. So thanks very much.
Right, so I hope you have enjoyed those presentations. I guess one of the main messages, as you can see, and this is the case also for those projects that are not being exposed today, that you did not see the videos of today, that we have a breadth of subjects really within the department, which is extraordinary, and a breadth of technique as well, techniques that are really ranging from histopathology to the most intricate molecular pathology aspects and sequencing and other areas of genomics, as well as organoid research. So I hope you get really excited about those. And in fact, what we thought we would do as well is to give you a very brief snippet of what our current students are thinking about their work within the department. So we have one very, very short video where two of our current students, Blake, who's currently undertaking honors, and Carolyn, who's done honors in the department a few years beforehand and is currently doing her PhD. What do they think about working in the department? So I'll leave Zoe to show you this as well.
Hi, I'm Blake, an honors student at the Department of Clinical Pathology. Currently, I'm completing a research project investigating the role of somatic TET mutations in pheochromocytoma and paraganglioma.
Hi everyone, my name is Carolyn and I'm a PhD student in the Department of Clinical Pathology at the University of Melbourne. I completed an honours project back in 2016 under the supervision of Professor Frédéric Hollande, where I interrogated differential drug responses in fluorescently labelled metastatic colorectal cancer cells.
For my undergrad, I was a genetics major and I was really most interested by my cancer genetics lectures, which led me to complete an undergrad research project with the Rare Disease Oncogenomics Laboratory. And this experience cemented my passion for cancer research and really also enabled me to find a new interest in bioinformatics, which was kind of unexpected. And so due to this really positive experience, I decided that I wanted to stick with the same lab for my honours year.
Whilst honours was a challenging year at times, I ultimately found it really rewarding. It taught me both the analytical skills and the technical skills that I needed to become a capable and independent researcher. So following honours, I was fortunate enough to receive a PhD scholarship, and this allowed me to continue my work in Fred's lab. I'm now writing up my thesis, which excitingly will be submitted at the end of the year. And without question, the foundations that I built in my honours year really prepared me well for my PhD journey, and I feel confident moving forward as a research scientist as a career.
So the learning environment, the facilities and opportunities at the department are all really good. This year I was extremely fortunate to receive the J.E. Taylor Honours Award, which was a fantastic motivational and financial boost. Also, the UMCCI has a really welcoming and collaborative atmosphere, and everyone is really enthusiastic about cancer research. So I would definitely recommend considering the Department of Clinical Path if you're someone who's interested in cancer research.
So if you undertake an honours project in the Department of Clinical Pathology, you'll be exposed to some of the most cutting-edge techniques at the forefront of biomedical research, like organoid culture, multiplex immunohistochemistry, genomics, bioinformatics, and even more. It's really rewarding to apply these techniques to tumour samples and from real cancer patients and see this work develop into discoveries which may one day help improve patient outcomes. Hope to see you in the department soon.
Right, so we've heard about what our students think. So they seem to be having a good time, but I think the important point is that one of the points I think that Carolyn made is that we really have an emphasis on providing the right environment to give you all the foundations that you need for the different careers that you are then hoping to get towards later on. So whether that be in the case of Carolyn going towards a PhD, or in the case of others to go towards medicine or other types of careers. So I think that's really a great emphasis that we have in the department. So I'll just then move on very briefly to a few last slides before we get to open the floor for you to ask questions if you wish. Hopefully you can see my screen again. The idea here for me now is to have a quick word about what is coming next in terms of applications if you are interested in some of the projects from the department. So you know, because you are looking around in other places as well, that you have access to various websites to get a bit more information about the different projects that you might be interested in. I really engage you to really speak to supervisors. In a normal year, you would visit the labs and I would really engage you in those contexts to actually try and speak to students because the students will tell you the reality of what's going on in the labs that they're in. But obviously it's a bit more difficult to do this this year, so you'll probably talk to the supervisors. Some of them might actually allow you to contact the students independently from that. That's certainly the case for me if you want to talk to my students. Once you have, I guess, found a number of people that whose projects you're interested in, and you have talked to them, you can move on to submitting your preferences submit the online course application first, of course, but also to submit your project preferences through Sonia. In the context of a master's, I haven't got a very specific process here, but effectively once you contact the master's team, they will ask you whether you have found a supervisor as well. So you need to follow a similar process in terms of looking for the projects on Sonia, find an expert if you want to know more about the different labs, or on the MDHS research project database, and then contact the supervisors, try and strike a deal with the supervisor, and then you can apply for the master's with a project already secured, and the supervisor will vouch for that. The key dates for honours this year are on this slide. 31st of October, you need to have put in applications for the course. The round 2 will be in late January this year. There might be slight variations, but I think those dates are pretty much set now, because everything has to be, in any case, finalized before mid-February, where, as you remember, you will start your Biomed 4001 Introduction to Biomedical Research subject. I think that's probably really the important things that I wanted to say. So I guess really the hope is that you will find something in the department that is of interest to you in terms of our course, as well as in terms of our research projects, whether for honours or masters. If you want to ask any questions, you may have questions of any kind, please either raise your hand via the Zoom function, or type it in the chat, whichever of those two is more practical for you. Or you know, open up your video, and I'm happy to take any questions directly with the video. So I'll check on the chat first if there's anything, but now you can feel free to ask any questions you want.
Do you have any opportunities for first, second year undergraduate students?
Okay, so that's generally the response to that is very lab dependent. So yes, labs do take up students early on as summer scholarships or as summer placements, paid or unpaid, depending on how much money the lab can have. As also there is the Europe program that you may have heard about, whereby you can also have paid placements. So what I would encourage you to do, if you're in that case, is to contact individual lab heads once you have found, I guess, an area that you're interested in, and discuss it directly with them. So it certainly happens, but sometimes labs have 2 students already at the time, and so I might not be keen to take someone in. So as I mentioned before, if people want to think about additional questions, I guess the process is really to go on the website, unless you have already noted the details for the different group leaders, but to go on the website, get the email addresses, contact us, and then we'll organize to have meetings with you to discuss what you're interested in, what you would like to achieve, whether or not you have the I guess the transcripts, records that support the fact that you will be an eligible candidate for the department and for the course as a whole. They're really the key issues. But I guess one thing to also realize, and that is important, is the fact that the cancer research stream is present at various levels. Obviously, when you think cancer research, you think Peter Mac, but that there are a lot of excellent labs, including in our department outside of the Peter Mac as well. I can see a question popping up actually. Zoe.
So do you know the average WAM students have when applying for individual projects?
Yeah, so that's an interesting question, but it also varies a lot. So I guess, again, as we mentioned earlier on during the presentation, there is a WAM cutoff of 65 anyway. That's a prerequisite to get into the course. So any students in a normal year that falls below that will not be considered unless an exemption is asked. And so the exemption would really sort of have to be documented as to why that exemption should be granted. So it means that you have to convince the supervisor already of why you should be taken if you're in that case. So we're looking generally higher up than that. And then it really depends on how many students are interested, and it depends on how, I guess, the lab's reputation has drawn several students to apply or to discuss potential projects with them, and so on and so forth. So I cannot give a blanket response to that. It will vary from the probably high 60s to early 70s, all the way to high 80s, depending on the lab. So my lab, for example, usually we're lucky to have very good students who are interested in us. So we're looking whenever possible for students who are in the 80s. But please, if you, and I'll repeat it, I guess, because you may not have been here when I started. If your WAM is not necessarily that high, but you're still interested potentially in keeping the door open for a research career later on, do consider masters, because otherwise that third year, at least one, so the third year marks will be counted towards your PhD scholarship score when you apply for a PhD scholarship, and that might not be fantastic. So if you have a score that's below 80, I would really encourage you to think about the option of going for a Master's of Biomedical Science, for example, in which case the 2 years of the Master's is a very new and different environment. You have some coursework left, but you have a lot of research, and if really research is your thing, you will thrive and you will get an excellent mark, and that makes you extremely competitive again for a PhD scholarship, where you might not have been otherwise if you had just used your third year mark plus honours.
We have another question.
Yeah.
So especially during the current situation we're in, has there been news in regards to us returning to university, in particular since honours and masters require lab work?
Yeah, it's a very good question. So unfortunately, I can't give you a blanket answer on that. So the current honours cohort, to give you an idea, and masters cohort for that matter, they're back in the labs. They're back in the labs. They have to respect pretty much anything else that our staff has to respect and our PhD students have to respect, which is that we are operating at 30% capacity in the lab at all times, which means that we have established very clear rosters as to who can be in the lab when, but they can be in the lab and their honors are progressing. So I am, because I'm coordinating the honors course this year, I actually don't have an honors student of my own, but I'm coordinating all the other students, including the Peter Mac students, because they have to enroll through our department as well. And so we have students progressing well, except that obviously they have been delayed. So the situation for next year, I cannot see a closure of labs like we have had earlier this year happen again. I mean, I can't guarantee it, obviously, but it doesn't strike me that this would probably be necessary. The worst that could happen is if we have a wave again, that we have reduced access to some degree, but I would expect that the disruptions should be lesser than the ones that we've encountered this year, where nobody was prepared and we had to really play by ear as we went. So what I'm trying to say is that I guess you'll be able to go through the honors year or the master's first year anyway. The worst that can happen is that for some of the students, what we've done, for example, is to adapt part of the project so that they were spending more time writing the literature reviews when they were forced to stay at home a little bit, spending more time also doing some analytical work and learning biocomputational techniques. So you can always play with those different things as well. And certainly that's not specific to our departments. If there are disruptions, that will be the same for everyone. Okay, so I guess if there are no more questions, or if a question comes late to you, please find me out. I shouldn't be too difficult to find, so you can pop me an email and ask the question. There's no problem at all. I hope that some of you got really interested by some of those projects. Go back to the website, talk to us, and we'll be really keen to see some of you in the department next year. So in the meantime, I hope that the rest of your semesters go well. Look after yourselves, and get in touch whenever you are interested. And we'll probably close the meeting here then. Have a great day.